In this video, we are going to be looking at the remaining two macromolecules, proteins and nucleic acids. Let's begin with proteins. Chemistry of proteins. So proteins are mainly composed of carbon, hydrogen, oxygen, and nitrogen, and there is a trace of sulfur in some cases. The monomers of proteins is something called amino acids, and there are a total of 20 amino acids in the human body that makes up all the proteins that we have. Of the 20 amino acids, 9 of them are considered as essential. That means you cannot make them in your body, and the only way to get them is to obtain it through your food source. So here are the 9 essential amino acids. Typically speaking, these amino acids are kind of present in all the proteins, but there are some foods that are particularly rich in certain types of amino acids. So for example, phenylalanine, you can get it from avocado and beans. They're particularly high in phenylalanine. Things like tryptophan, it's particularly high in oats, spinach, so on and so forth. But generally speaking, if you are not a picky eater and you have a well-balanced diet, then you are going to be consuming a sufficient amount of these essential amino acids, and you don't have to worry about any negative health impacts. There's a rare genetic condition called PKU, which stands for phenylketonuria. In PKU, the individual cannot metabolize phenylalanine, which is one of the amino acids, and that could lead to dangerous buildup in the body and in the brain, which can interfere with brain development. So if you do a blood test and you determine that the child has PKU, then they need to be put on a special diet. They sometimes have to consume these PKU formula, which ensures that the child is going to get adequate proteins in their diet without taking in phenylalanine. Here is the general structure of an amino acid. As we mentioned earlier, there are 20 amino acids that makes up the protein, and they all share this general structure. So there is a central carbon here, and the central carbon is connected to a hydrogen atom, as well as this group here called the amino group. And then over here, it's connected to something called the carboxylic acid group. Okay, so I'm just going to put acid here. That's why they're called amino acid, because of these functional groups that are attached to the central carbon. There is going to be an R here. The R stands for side chain. And depending on what side chain is connected to the C, that's going to determine the identity of the amino acid. So here you can see there are the 20 amino acids that makes up all the protein, and there are going to be different side chains in these molecules. So it's a little bit difficult to read because we haven't talked about how to look at condensed chemical structures. You eventually will do that in your chemistry course. But for example, there's a ring here in one of the side chains, and over here you can see some side chains are going to carry a negative charge. Some of them are going to be acidic, some of them are going to be basic. So each amino acid is going to have its own property, and depending on which amino acid you use to build your protein, that's going to determine the property of the protein as well. So how do you create a bond between two amino acids to link them together? Just like what we learned in lipids and what we learned in carbohydrates, we are going to use dehydration synthesis to connect two amino acids together, to connect the two monomers together. So you might remember this is a general structure, right, with the central carbon, and then there is the acid group. And over here we have the central carbon, and then there is the amino group. So what happens is you can take an OH from the acid group, and you can take an H from the amino group of another amino acid, and after you remove them, you're going to form water. And by removing the OH and the H, the carbon over here is going to be able to form a bond directly with the N, and this new bond is shown here. This is called a peptide bond. Okay, peptide bond. So by joining two amino acids together, you form a dipeptide. Okay, two amino acids. So This can continue to go on. This end here, this acid end OH, you can remove the OH here, and then you can put another amino acid here, and then here we have the amino group, and then the acid group here again. So I can remove the H from an amino group of another amino acid, take out the H2O, then you will be able to create another peptide bond. Okay, so in a protein, there is going to be one end that is going to end with the amino group. So we call this the N terminus. And over here we're going to have another end that is going to end with the acid end. So we call that the C terminus. Okay. When it comes to the structure of proteins, there are actually 4 levels of protein structure. There's primary, secondary, tertiary, and sometimes some proteins will even have a quaternary structure. Let's take a look at what all these level means. First, the primary structure of a protein. The primary structure refers to the unique amino acid sequence in a polypeptide. So here you can see this is the N terminus and this is the C terminus, and these circles represent the different amino acids. So if I were to read the sequence, so for example, this could be valine connected to methionine connected to alanine, and then another adenine, for example, and then we just keep on reading the whole sequence. That unique sequence is what we call the primary structure of the protein. And this order of sequence is actually determined by your DNA. In lecture 6 and 7, we're going to learn how your DNA codes are going to determine the identity of the amino acid that goes into building your protein. And remember, each of the adjacent amino acids are going to be linked by a peptide bond. And you can create a peptide bond by removing a water molecule from 2 amino acids. So after forming the primary structure, the next level is the secondary structure. It turns out this linear polypeptide is going to start to fold up into various twists and turns, and depending on what amino acids are in a particular segment, that's going to cause it to either fold up in these things called alpha helix, or it's going to cause them to fold up in these parallel sheets called beta pleated sheets. Okay, so let me try to draw this out for you again. This is your linear sequence, your primary sequence, and then afterwards some section might fold up into these alpha helices, and then other sections they might form these zigzag beta pleated sheets. So whether it forms these alpha helices or the beta pleated sheets depends on the amino acids that are found within a particular segment. And the structure, the helices and the beta pleated sheets, they are actually stabilized by what's called hydrogen bond. Okay, so we first learned about hydrogen bonds when we talked about how water molecules are attracted to each other, right? So similarly, over here, we have a carbon and a double bond connected to the O. That makes the oxygen slightly negative, and over here we have the nitrogen and the hydrogen, and hydrogen is slightly positive, so they are gonna be attracted to each other, and that interaction is called an hydrogen bond. So with all these hydrogen bonds, you can maintain the structure of the alpha helix, and similarly you can see the hydrogen bonds here holding the beta pleated sheets together. So this is a ribbon model, like a nicer one than what I have here. Usually they use these arrows to represent beta pleated sheets, and then the ribbons, the coils are going to be the alpha helix. In mad cow disease, there is a protein called the prion proteins, P-R-I-O-N, and the normal prion proteins, the PRPC, that's what the normal one is called. There are the 4 alpha helices in the protein structures. And in mad cow disease, what happens is the protein is going to be misfolded into something that looks like this. So instead of having 4 alpha helices, you now only have 2 alpha helices, and you have 2 beta pleated sheets. And what happens is when you have a lot of this misfolded version of the protein, they start to aggregate together, they start to clump together, and they start to accumulate in your brain. And when that happens, it will cause neurons to die, and it will leave behind big holes in the brain, which is ultimately what causes the pathology of the disease. Next, we have the tertiary structure of a protein, which basically refers to the overall three-dimensional shape of the protein. And how the protein folds up and what kind of shape it has actually depends on the interactions between the side chains, the R groups of the different amino acids that makes up the polypeptide. So depending on the properties of these side chains, you might remember some are positive charge, and that might be attracted to the negatively charged side group in another part of the protein, and therefore allowing them to create a connection that will reinforce the structure of the proteins. And depending on what interactions exist within the protein, the shape of the protein will be different. So here you can see in this diagram, there is the yellow ribbon here that represent the polypeptide backbone. And the polypeptide backbone is basically the repeating structure that you get when you connect the various amino acids together. So here you can see this is the first amino acid, right? R1 represent the side chain of the first amino acid. There is the central carbon. This is the N-terminus, and you can see the acid end of this amino acid is going to form a peptide bond with the amino group of the next amino acid. And then here you can see R2 is the second side chain. So even though the side chains are going to be different, because you're going to have different amino acids being connected together, the backbone remains the same. It's just repeating unit of NCC, 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 and then it just kind of keeps on going and going. So this yellow part, it represents this backbone. Coming out from the backbone would be the side chains. Okay, so different side chains have different properties, and some are going to be hydrophobics, and they tend to come together. So if one part of the protein has a hydrophobic side chain, and then another part here also has a hydrophobic side chain, then these two parts might tend to come together and causing the protein to create a bend like this. There's something called a proline kink. Proline, it's a special type of amino acid that has a kind of like a ring structure to it, and it turns out if you have multiple prolines next to each other, it will create a sudden bend in the proteins. There are these hydrogen bonds. If you have a side chain that is going to create a slightly positive hydrogen because of uneven electron distribution, that's going to be attracted to the slightly negative oxygen in another part of the proteins. You don't really have to memorize all these interactions. This is just to show you that if the side chains have different properties, they're going to create different interactions, and these interactions are crucial in determining the overall 3D shape of your protein. So a lot of proteins will stop at the tertiary structure. However, some proteins will go an extra step and have these quaternary structure. Some proteins are actually made up of multiple polypeptides, multiple protein sequences, and they need to come together to form an overall functional protein. So this is an example of such protein. There are two pink polypeptides and two purple polypeptides. Each of these individual polypeptides will have their own primary, secondary, and tertiary structure, and the quaternary structure refers to how they come together to form this big overall protein. So this is just a summary of the 4 levels of protein structures. Let me just recap the whole thing for you. The monomers for building proteins are amino acids, and there are 20 different amino acids. Here you can see the general structure of the amino acids: central carbon, amino group, carboxylic acid group, and then there's a side chain. So what amino acid connects together to form the protein is going to be the primary structure of the protein. So if you were to read the individual amino acid in the order that you link them together to form this long chain of protein, that will be the primary structure of the protein. And after you link them together, depending on the type of amino acid you have in a particular segment, they will either fold up into these alpha helices or beta plated sheets. Okay, so whether you have helices or sheets, that's going to be the secondary structure of the protein. Afterwards, the overall three-dimensional shape of the protein is going to be determined by the side chains that you have in the amino acids. And depending on how the side chains interact with each other, the overall shape is going to be different. And most of the proteins will stop at the tertiary structure. However, in some cases, two or more proteins, polypeptides, would come together and form an overall structure complex that has a different function, and that is considered as the quaternary structure of the protein. So don't think of each level of the protein structure as independent of each other. In fact, each level of the structure helps contribute to the overall shape of the protein. The primary structure will cause the helix and the plates to form, which then would continue to fold up into the three-dimensional shape. So one level leads to the next. They kind of scaffold on top of each other. And the overall shape of the protein is called the conformation of the protein. The conformation of the protein is very important for its function. Typically, if you change the shape of the protein, you're going to end up causing the protein to lose its function, or it's going to cause it to be functioning in a different way, and typically that's not a good thing in terms of bodily function. When we break down the protein, we say the protein has been denatured. Okay, and if you denature the protein, you're basically destroying the shape of the protein, destroying the conformation of the protein, and like I said, it's going to affect its function. So when you fry an egg or cook the meat, you are basically denaturing the protein. So the egg white, which is where the proteins are, by the way, looks very different when it's cooked than when it was raw, and there's no going back. Okay, and that's also true for many proteins in the body. Once you denature them, it's very difficult and almost impossible for them to be undenatured. Okay, it could happen in some rare cases, but typically speaking, once you denature it, it's a one-way transformation. Like you cannot uncook your meats, right? For example, so there are various factors that's going to cause a protein to denature. Temperature, okay, not just high temperature. Low temperature can cause proteins to become denatured or misfolded as well. If you think about Jello, right, by putting the gelatin liquid in the fridge, it's going to basically solidify, right? And that's an example where low temperature can also cause the protein to misfold. pH, we talked about pH earlier in the lecture. If you change the acidity of the environment that the protein is in, it's going to cause it to misfold. Changing the salt concentration is also going to disrupt the protein shape. Let me just go back a few slides here. Because some of these interactions are held together by ionic bond, and you might remember ionic bonds are very sensitive to ion disruption. So for example, if I add NaCl, sodium chloride, to the solution, then now the Na is going to bind to the O minus here, and the Cl is going to bind to the NH3 plus here, and therefore it could break this ionic bond, causing the protein to unravel, to fall apart. Right? So that's why changing the salt concentration is going to disrupt the shape of the protein. Also, the type of solution is going to matter. If you switch a protein from an aqueous solution, a hydrophilic solution, to a hydrophobic solution, then you might cause the protein to flip inside out. So don't worry too much about how the factors affects the shape of the protein. Just know that these factors will affect the shape of the protein, and by changing these factors, you're going to cause the protein to misfold, and ultimately it's going to cause it to lose its function. Here is an example of how changing the primary sequence of a protein will change its structure and therefore resulting in a loss of function. This is what a typical normal red blood cell looks like. It's kind of like a jelly-filled donut with a little pinch in the center, and inside your red cell you have something called hemoglobin, which is important for transporting oxygen. So hemoglobin is a protein with quaternary structure, which means it's made up of multiple polypeptides, and in one of the 4 polypeptides, it has 146 amino acids. Okay, so the primary sequence of the normal hemoglobin starts with valine connecting to histamine, leucine, threonine, proline, glutamic acid, and so on and so forth. If there is a mutation in your DNA, the instruction that tells you how to build the protein, if there's a mutation, if there's change in the DNA, then the instruction is going to change. And if that mutation causes number 6 to change from glutamic acid to valine, just a single change in the amino acid sequence of the hemoglobin will cause your red cell to look like this. Then you end up having something called sickle cell anemia. And in sickle cell anemia, because the shape of the red cells are very different than the normal one, they are not very good at carrying oxygen, and in fact they are going to get stuck in blood vessels, and that could cause some pain in some cases, and so you won't be able to deliver oxygen effectively. You will feel tired all the time. You're going to have shortness of breath, for example, and so all that happens because you change a single amino acid in a 146 amino acid protein. Okay, so this illustrates how important it is to have the correct primary sequence in the protein, because ultimately that's going to determine the overall 3-dimensional shape of that protein. Functions of proteins. So proteins can be used as structures in the body. So muscle is definitely made up of proteins. There are 2 key proteins in muscles, something called actin and something called myosin. You're going to learn that in your anatomy course next semester. You have proteins that makes up harder structures like nails, horns. We don't have horns, but animals, their horns are made up of proteins as well. Hair, spider webs, these are all proteins. Proteins also makes up hormones. Not all hormones are proteins in the body. Some are lipids. We talk about how estrogen and progesterone are steroid hormones that are made from cholesterol, but there are other hormones in the body that are made of proteins, such as insulin. Okay, so insulin is something that is secreted by your pancreas, and you need that for regulation of blood sugar level. We can also use protein as an energy source. Is not the preferred energy source. The preferred one is glucose, and if you're running low on carbohydrates, then you can use lipids. But then if you're really desperate, then you can use proteins as your energy source. And there's something called enzymes. Proteins form enzymes in the body. Enzymes are basically biological catalysts that would speed up chemical reaction. Okay, a catalyst is basically something that would help speed up a chemical reaction without being used up itself. So and enzymes are proteins that will catalyze chemical reactions in living things by lowering something called the activation energy, or E subscript A. So in any general reaction, you will start with some reactants, some ingredients, and that's going to be converted into some products. But in order for you to kickstart the reaction, you must always supply a little bit of starting energy. Okay, so the energy that needs to be supplied, the energy that needs to be supplied to the reactants in order for the reaction to occur is called the activation energy. Okay, so in many reactions, the activation energy is actually quite high, and within a biological system, it might not be possible to supply that energy. Okay, and even if you're able to supply that energy, it would take a very long time for the enough reactants to go to get that energy and to be converted into products. So to speed things up a little bit, we have these catalysts, we have these enzymes within the body. So the blue line here shows you in order for the reactants to be converted into products, you must give this much energy. Okay, so in the presence of the enzyme, you only have to give this much energy. So by lowering the activation energy, more reactants will be able to be converted into products, and therefore you're going to be able to make this process a little bit faster. Okay, it's like the cutoff to go into your next program is going to be 90%. Okay, the marks that you need to go into the next program is 90% average. Okay, so not many people will make it. So with the help of an enzyme, now the cutoff is like 65%. Okay, then a lot more people will be able to make that cutoff and be converted into products. That's just like a loose analogy of what an enzyme does. So here is a closer look at how enzymes actually work. Okay, so enzymes are proteins, and they have a special site called the active site. So here you can see this blue diagram is going to have an active site. Now the active site has a special key and lock relationship with its substrate. Okay, the substrate is the target that the enzyme is going to work on. Okay, so in this case, we have these cubes, and then we have this whatever shape it is, this yellow thing. Okay, let's say the red cube here is glucose, and let's say this yellow thing here is galactose. Okay, so what happens is if I don't have an enzyme and I wait for glucose to combine with galactose to form, say, lactose, that will take a very long time. Okay, because the activation energy for that reaction is going to be quite high. In the presence of the enzyme, the active site will allow the glucose and the galactose, your reactants, your substrate, to bind into the active site, and then it will lower the activation energy and help you form the product much faster. Okay, so in this case, the glucose and the galactose will go into the active site, and then the enzyme will perform the dehydration synthesis reaction, and you will get the product. So this product is going to be lactose. Okay, so similarly, you would have a different enzyme for the reverse reaction. Okay, this time you would have the lactose, which acts as the substrate now, will go into the active site, and now the enzyme will use hydrolysis to split the bond between the glucose and the galactose, and then releasing them. Okay, so the point is, each enzyme is going to have a particular substrate associated with it, and you can see if you change the shape of the active site, then the substrate will no longer be able to fit. Okay, so if there is a mutation, or if there is high temperature that causes the enzyme, the protein, to denature, to change the shape. Let's say this changes to like a semicircle instead, then the cube will no longer fit, and it will no longer be able to do its job. Okay, so enzyme is an example of protein function within the body. Sources of proteins. Meat products are excellent sources of proteins, and as mentioned earlier, if you have a well-balanced diet, then you probably will be able to consume all the essential amino acids in your regular diet, and there's no need to focus on eating a particular type of proteins to boost your amino acid intake. It's kind of all over the place anyways. And if you don't eat meat, if you're a vegetarian or vegan, then excellent source of protein include beans, soy product, nuts. They're all rich, very rich in proteins. So that's it for proteins. Let's now talk about nucleic acid. So nucleic acid is going to be made up of carbon, hydrogen, oxygen, nitrogen, as well as phosphorus. Okay, so the monomers for nucleic acid is something called a nucleotide. Okay, so we're going to look at the structure of a nucleotide in the slides coming up. But first, there are two main types of nucleic acids in the body. Right? The first one is probably something you have heard of before, deoxyribonucleic acid, which is what DNA stands for. And then this one is a little bit less common to people. It's called the RNA, which stands for ribonucleic acid. So these are the two types of nucleic acid in the body that plays very, very important function, as we will see in the coming up lectures in the course later. So the nucleotide, the monomers, the building blocks for nucleic acid has three components to it. There is a phosphate group. Phosphate is PO4 -3, something that you probably will learn in chemistry class later, and it carries a negative charge. Okay, so this is something that's important to remember. It's going to come back later on in the course with some significance. There is also a 5-carbon sugar. So if you think of glucose, something that we talked about earlier, glucose is C6H12O6. So this would be a 6-carbon sugar. Okay, but the one that you find in nucleic acid, it's a 5-carbon sugar. Okay, so there are two versions of the 5-carbon sugar. It's either a deoxyribose sugar or just simply a ribose sugar, and I'll tell you the difference between the two again in a few slides later. Okay, and lastly, the last part that makes up a nucleotide, it's a nitrogenous base. Okay, and there are different versions of nitrogenous base that you can have in a nucleotide as well. So 3 parts to the nucleotide: phosphate group, ribose or deoxyribose sugar, and then there is the nitrogenous base. So here is a picture of a typical nucleotide. You have the phosphate group here, the PO4, right? So that's phosphate in the center and 4 oxygen, and it carries a negative charge. And this is going to be connected to a sugar, a 5-carbon sugar, and depending on whether it is a nucleotide for DNA or if it is a nucleotide for RNA, the 5-carbon sugar is going to be different. Okay, so in DNA, the 5-carbon sugar is a deoxyribose. Compared to in the RNA, it is just a ribose sugar. So the difference basically points to this carbon. Okay, this carbon right here. Okay, if the carbon connects to an OH group, then it's a ribose sugar, and if the carbon connects to just the H, then we call it a deoxyribose because it's missing the oxygen, right? So that's what the D stands for, right? In DNA, the deoxyribose nucleic acid. Okay, so the nucleic acid can either have the deoxyribose sugar or the simple ribose sugar. Okay, so the third part of a nucleotide is going to be a nitrogenous base. It's called a nitrogenous base because this contains nitrogen atoms in them. So the nitrogenous base for DNA can either be something called A, G, C, or T. Okay, so each of these letters actually stands for something, and we'll look at that later on. And these are the 4 genetic codes that makes up your entire genome. Okay, that basically forms the blueprint of life. And over here on the RNA, the nitrogenous base can be A, G, C, just like the DNA, but instead of having thymine, T, in the RNA, we have something called uracil instead. Okay, so there are 2 fundamental differences for the nucleotide between DNA and RNA. Okay, just to recap, it's going to either missing an oxygen, right, in the deoxyribose, or having the oxygen in the ribose sugar. And for the nitrogenous base, you either have thymine, okay, as the fourth base, or you have uracil as your fourth base in RNA. The nitrogenous base, what they stands for is written here. So, for example, the A stands for adenine, right? G stands for guanine, C stands for cytosine, T stands for thymine, and U stands for uracil. Okay, and remember, the uracil is RNA only, whereas the thymine, that is the DNA only. Okay, and they are further divided into two groups. Okay, the nitrogenous base is either classified as a purine, which means it has two rings in its structure, or it's classified as a pyrimidine. Okay, pyrimidine only have a single ring in its structure. Okay, so purine is adenine and guanine, whereas pyrimidine is cytosine, thymine, or uracil. Now there is a condition called gout. You might have heard of it, or you might actually have gout. This tend to be common in people who are a little bit older. And what happens is in gout, your body cannot get rid of uric acid crystal, and they start to build up in joints, particularly near the big toe, and it causes swelling and extreme pain. And it turns out if you have gout, and you consume purine, purine is the adenine and the guanine, right? And that's going to actually make the symptoms much worse. Okay, so there are certain foods that are particularly high in purines. You can see them in the pictures here. So seafood, red meat, alcoholic beverage, they are all quite high in purine, and you should not be consuming those if you have gout. So just to take a look at the structure of DNA, we're actually going to have a whole lesson on DNA later on in the course. So now this is just going to be a brief overview. You can see the DNA molecule is a double helix, okay, which means there are two twisted strands here, and the backbone of the helix is made up of the sugar and the phosphate. Okay, so the yellow here is the phosphate group, and then the blue here represent the sugar. Okay, and in DNA, the sugar is the one that's missing the oxygen, right? The deoxyribose, and sticking into the middle is going to be the nitrogenous base. Okay, so you can think of the DNA structure as a twisted ladder. So here you have your regular ladder, okay, and the sides that you kind of hold on to, that would be the sugar phosphate backbone, okay, and then the rocks in the middle would be the nitrogenous base. Now there is something called base pairing rule. It turns out the adenine will always be pairing with the thymine, whereas the guanine will always be pairing with the cytosine. Okay, so as you remember, the A and G, they are the purine, which means there are two rings in the structure, whereas the T and the C, they are the pyrimidine, which means they have a single ring in the structure. So whenever you have a purine binding with a pyrimidine, then the space across is going to be 3 rings. Okay, so you can see here is the adenine, here is the thymine, and it's 3-ring structure, and here is this cytosine, 1 ring, combining with the guanine, which is 2 ring, and that's important because you want to make sure that the distance between the two backbones are constant going through the entire length of the DNA. That kind of constant length is going to add to the stability of the DNA structure. If you take a closer look at this diagram, you might notice that one side of the DNA is actually upside down compared to the other side, and that's another special property of DNA. Okay, not only are they parallel, but they are anti-parallel, meaning they run in opposite direction. Okay, and there is a reason for that, which we'll get into in later lectures. Okay, so because they always follow the same base pairing rule, A goes with T, G goes with C. The two strands of a DNA are called complementary, meaning if you know one side of the strand, you can predict what's on the other side. So if I have one strand here, right, written here for you, and I ask you to predict what's on the other strand, it should be very easy. All you have to remember is A pairs with T, okay, it spells the word at, right, that's how you can remember it, and then G pairs with C. Okay, so that means if one side is A, the other side must be a T, and G goes with C, G goes with C, and then C goes with G, T goes with A, A, and then A goes with T, T, A, T, G, C, and C. Okay, so anytime you see an A, you know it's going to be pairing with a T. Anytime you see a T, you know it's going to be pairing with an A, right? And same thing goes for C and G, right? So if you know one strand, you will always be able to predict what's on the other strand, and that is what we call complementary. Complementary. Here is another question related to the complementary rule of DNA base pairing. Let's say we have a cell that has 40% adenine. Okay, what would the percentage of the other three nitrogen base be? Okay, so let's think about it. We know that A always pairs with T. Okay, so if you have 40% adenine in a cell, that means there has to be 40% of thymine as well, right? Because the number of A's should be same as the number of T's. They always bind to each other, right? So a total is 100%, and if you take 40 plus 40, that's going to give me 80%. Okay, so the A's and the T's are going to take up 80% of all the bases, which means if I take 100 minus 80, then I get 20%. Okay, 20% would be the remaining base for C and G. Okay, and so if you divide the 20 into 2, you will know that there is going to be 10% C and 10% G. Okay, so try to go through this example yourself again, and if you have any questions, we can talk about it during the tutorial section. Next, we have RNA. Okay, RNA is different from DNA, not only because of the type of nucleotides they have, but also it's because they are single-stranded. Okay, so DNA is the double helix. RNA, you only have one side of the ladder. The backbone is also made up of the sugar phosphate backbone, but you should know by now that it is a different type of sugar, right? It's the ribose sugar, the one with the extra oxygen, compared to the one you have in DNA. And the nitrogen base, there is the adenine, cytosine, and guanine, just like DNA, but uracil replaces thymine, right, in DNA. Okay. There are three types of RNA in the body. There's something called messenger RNA, which will help as a copy of your genetic code. There's something called ribosomal RNA that helps with the protein synthesis process, and then there is something called transfer RNA, which also helps with the protein synthesis process. So we will go into a lot more detail in terms of the role of these individual RNAs later on in the course. Here is a big summary of everything we've talked about. Okay, you can see carbohydrates, right? We talk about starch and glycogen, and then the monomers for those things would be glucose. Functions is storage of energy, right? It's a good table to go through yourself. You don't have to memorize the examples in this table necessarily, but it should reinforce some of the things that we've talked about for the lecture. So that's it for lecture 2. It's a very long lecture, but I hope you did learn something from it, and please work through the study guides. Those questions will definitely help you prepare for the quiz and tests that are coming up. Thank you for watching.